bacterial cell bursting and releasing all of the phage. So they replicate incredibly quickly. In the time it takes one bacterium to become 200 bacteria, one phage can become 10 to the 16 phage. So the rate at which they expand is just incredible. And we can use those in medicine. So we've isolated phage against most of your favorite bacteria, most of the pathogens, most of the bacteria that cause problems in hospitals, Staph aureus, Salmonella, Acinetobacter, Listeria, Campylobacter. We've been able to isolate phage against all of these. And of course, what you might like to do is use these then as very narrow spectrum antibiotics to treat a particular disease. And they've been doing this in Russia for 100 years. And they have lots of different phages that they use in Russian hospitals, but it really hasn't transmitted to the rest of the world. But we're beginning to catch up a little bit. So here's an experiment we did, and I just want to talk you through this one. We deliberately infected these mice that you see in these pictures with Pseudomonas aeruginosa. And Pseudomonas aeruginosa can cause lung infections. And you can see, I hope, that the mice are infected in their lungs. We're using a trick, a light-emitting Pseudomonas, so we can look and see the infection through the body of the living animal. And so you can see the infection beginning to build in the animal across that time period. There's two groups here that we're showing with two different Pseudomonas infections, but in both cases, the lungs become severely infected, and then the gut starts to become infected, and this would be a lethal infection for these animals. And this, of course, happens in humans as well. We isolated two bacteriophages that you see on the top left for Pseudomonas aeruginosa. So these are bacterial viruses that kill Pseudomonas. And firstly, we showed that they could kill the bacteria when they form biofilms on lung cells. So that's on the bottom right of the slide as you look at it, or the bottom left of the slide as you look at it, sorry. And so then what we did was each of these mice, we put the virus, the bacteriophage on their nose and let them just inhale the virus. And within 8 hours, the infection was almost completely eliminated. And this is because, of course, these bacteriophage can find their target and then multiply within the animal and kill all of the Pseudomonas that are present. And all of these animals survived. So this is a life-saving treatment based on these bacteriophages. And now people are starting to use these in hospitals outside of Russia. Here's a study that was done in Europe, again on Pseudomonas aeruginosa, using a cocktail of these Pseudomonas phage. I'm not going to go through it in any detail. You can read the paper if you're interested. But basically, it managed to eliminate the infection almost as well as the best antibiotic. Not quite as well, but almost as well. But of course, the good thing is it's not killing anything else. The antibiotic is killing lots of bacteria and causing damage to your microbiome. The phage is only killing the Pseudomonas aeruginosa. Here's another example of a case report. 3 people in this study were administered phage for Enterococcus faecalis infections, antibiotic-resistant Enterococcus faecalis, and basically the phage worked in all 3 patients. Now, I understand 3 patients is not enough to decide medical practice or to change medical practice, but it's just giving us a hint as to why they use these treatments in Russia and why they apparently believe that they're so successful. And here's another example where they used bacteriophage to treat a patient with Acinetobacter baumannii infection, a very hostile infection, antibiotic resistant, had spread through the body. This patient was in a coma, was near death. They administered the phage. They could recover the phage from the patient, and the patient made a complete recovery. In fact, this is the patient here that you see on the left, Tom Patterson, who was treated with this Acinetobacter baumannii phage and made a complete recovery. And he now travels the world telling people about the benefits of phage. You can use phage for burns. That's an example of a patient in France receiving phage for a Pseudomonas or Staph aureus infection and a burn. And they've done them in England for otitis media, for ear infection. So more and more people are starting to turn to phage, and I think pretty soon we'll see the first random clinical trials, the first RCTs to prove that these really can work. So bacteriophage are a kind of a pharmabiotic. They're not living, they're not bacteria, they come from bacteria, and they can have an impact on host health. Now, we use an expression in English, the elephant in the room, which is the problem that nobody acknowledges. And the problem with bacteriophage is, of course, that cells become resistant very quickly to them as well, just as cells become resistant to antibiotics. But there are some ways we can overcome that. We can use careful selection of phage cocktails. Maybe we don't have to eliminate the bacteria, we only have to reduce it to a level where it's not causing any problems anymore. Or perhaps we could use some enzymes from these bacteria phage, not the whole phage itself. And here's just a very quick example, again from our own lab, showing that here we've infected animals with Staphylococcus aureus. Here's a skin infection on the bottom and a nasal infection on the top. And when we add one of these phage enzymes, you can see that within seconds we can eliminate the infection using these phage enzymes. So these truly are pharmabiotics. And these have been used in humans as well. So here's an example where they used endolysin to prevent death from normally lethal vancomycin-resistant enterococci infections. Another very important pharmabiotic are bacteriocins. Now, these are small molecules produced by bacteria which kill other bacteria. And so they can be very useful as kind of, again, as antibiotics. They're heat stable, there are lots of different types, they're very complicated, and they're used commonly in food to preserve food so that we've eaten them for many years without any negative effects. We showed in one study, and this is quite a complicated slide, but we showed in one study that a probiotic producing a bacteriocin could protect mice from a lethal infection of Listeria monocytogenes. So if you look at the animals on the bottom left, you can see that we treated these animals with Listeria monocytogenes, and then we treated them with a probiotic, 6 different probiotics. Only one probiotic worked. The one you see there, Lactobacillus salivarius UCC 118. That was the only one that could protect the animals against infection. And when we looked, we found that UCC 118 produces a bacteriocin. And that bacteriocin kills Listeria. So we did the obvious thing. We made a mutant of that bacteria which couldn't produce the bacteriocin anymore. That's the UCC 118 BAC negative. And when we use that, and if you look at the image on the right, you can see mice on the left with no probiotic, they become infected. Mice in the middle who get the probiotic are uninfected, but mice on the right who get the probiotic that cannot produce the bacteriocin, once again they're infected. So here we really have a probiotic effect which we can link to just one single molecule, the production of this bacteriocin. We can use bacteriocins in animal medicine. Here you see an example of mastitis in cows. Mastitis is a very big problem, of course, in humans. It's a very big problem in animals as well, in cows. And here what we did was we introduced a bacteriocin into the teat of cows, and then we deliberately infected the cows with Streptococcus dysgalactiae to cause mastitis. So this is a very big experiment to do on very large animals. And what we saw was when we infected the animals that got no bacteriocin, these are the ones up here, then 50% of the animals got infected. About 20% of the animals had the bacteria in the teat, but they weren't infected or they weren't showing symptoms, and only about 33%, 34% remained healthy. But when we included a bacteriocin in this preparation, then 90% of the cows were healthy and did not carry the organism. 10% carried the organism even though they were healthy, and none developed mastitis. So we could show that these bacteriocins can be used almost in the same way we would use antibiotics. But they're very narrow spectrum. They don't kill everything as antibiotics do. Here's another example, again from our own lab, showing that here we found a bacteriocin, which we call thuracin. It's quite a complicated molecule produced by these genes here. And this bacterium kills Clostridium difficile, which causes, of course, a major infection in hospitals. It's Clostridium difficile-associated diarrhea, which can often be fatal, unfortunately. And normally the treatment for CDAD is antibiotics. So CDAD is triggered by antibiotics and then treated with more antibiotics. So, of course, the damage to the microbiome is enormous. And we show that over here. If we take a fecal sample which has been deliberately colonized with Clostridium difficile, then it grows within that sample. If we add antibiotics, it kills this Clostridium difficile, which is good, but it causes major damage to the microbiome. So the microbiome changes very dramatically. But if we add thuracin, this narrow-spectrum bacteriocin, we can control the Clostridium difficile without causing any damage to the microbiome. And I think in future, we will look back with some kind of horror at the way we used to use antibiotics in medicine, without any regard for what it was doing to the microbiome. I think clinicians in 20 years' time, maybe in only 10 years' time, will be really reluctant to use these broad-spectrum antibiotics because of the consequences for the microbiome. And we're going to need alternatives like bacteriocins and bacteriophage. The last thing I wanted to mention was bacterial biomass. What if you introduce heat-inactivated bacteria? Bacteria which have all of their molecules, but which are no longer living.